So I've gone ahead and written up here a little bit more precisely what parts of the textbook you could look at for, to reinforce what we go over in lecture. But again, the main thing you should focus on is the material in the lecture. But I use, like today, you'll see that, we go through all the figures that are in this chapter one by one to try to explain this to you. So you can use the textbook quite a bit to reinforce what comes in lecture. But I'll try to put up something like this each lecture so you know specifically which pages to be focused on. All right. So we talked we talked on Wednesday about the idea that living organisms encode some sort of instructions in the cells that allow them to replicate, whether it be things like bacteria dividing and making two identical bacteria, or something like mammals where you have mating and then you get offspring that more or less resemble the adults. Again, so like in the example of bacteria, where you can start with a single bacterial cell and end up with this large mass very quickly because they divide quite rapidly and they divide by fission, so they just enlarge and they split in two, right? But it's not like every protein in that bacteria just makes a copy of itself, right? So somehow there's some sort of information being passed along that's in both of these daughters that tells it how to keep making the constituents of that cell. And likewise, if we think about, say, a vertebrate embryo, so if I showed you a single cell embryo, it would look pretty much the same whether it was to become an elephant, a human, or a butterfly. Okay, pretty similar looking one cell stage, but obviously there's some sort of information in that egg that has a different outcome then. Okay, and so what we talked about was trying to figure out historically what that information was. And so one way to approach this was to look at instances where the information got changed. And an example that we talked about was bacteriophage. So bacteria normally, of course, are dividing and making more bacteria to making more of the components that they need for themselves, the characteristic proteins and so on that are in that cell. But if they get infected by a bacteriophage, that phage comes along, sits down on it, it actually then comes off, but that bacterial cell, instead of making more bacteria, now it actually switches over just to making more viruses. And so its instructions in some way are changed so that it ends up just making a bunch of phage. Okay? And so one of the questions was, what was the material that was carrying this information? And in fact, most people actually thought it was protein, that somehow, because it was more complicated and stuff, that was the material that was being passed along that had the information in it. And so there was this experiment that we went over by Hershey and Chase from 1952. And the idea here was relatively simple. So what they did is they allowed the phage to attach, and they had the phage either labeled with radioactive sulfur or radioactive phosphorus. In theory, you could have done both labels at the same time if you could tell the difference between the two radioactive compounds. And then they would let the phage attach just for a minute, okay? And then they would put it in a blender, and the blender would knock the phage off, okay? But these bacteria, if they let them sit and grow, they were infected, and they would die and just produce phage particles. So they were already irreversibly changed. And so what they wanted to know is if they labeled the DNA, in this case, by using radioactive phosphorus, or if they labeled the protein of the phage, in this case, using radioactive sulfur, which was actually in the bacteria, okay? And so what they could do is centrifuge this. After blending it, they could centrifuge it, and the bacteria would pellet down at the bottom, and the phage that they knocked off would stay up in the liquid and supernatant, okay? And so if the answer is that it's in that DNA, right, then what would you expect to have happen? Okay, and we can try, let's see if I can try to do this with a clicker. It's not guaranteed to work, but we'll give it a try. Okay, so the idea is that, let's put that question back over there. All right. Okay, so what we're doing is the DNA is radioactive with phosphorus and the protein is radioactive with sulfur, and let's assume for the moment, which is the right answer too, that it's in the DNA. So where would the radioactivity end up? Okay, and I guess I can start this thing and let's see if it works here. What's that? Let's assume that the answer is that it's going to be in the DNA. So I think this is working. I guess I'll give you about, let's say, 20 seconds. All right. Okay, everyone voted? Okay, so now let's see if I can figure out how to find the answer. Okay. All right, so most of you answered D, okay? So, and that's right, okay? So the logic behind this is that what happens here, okay, what happens here is if it's in the radioactive phosphorus and that is the material that's being transferred into the bacteria, then when you centrifuge it down, the radioactive phosphorus is in the bacteria, so that's P32 in the pellet where the bacteria ended up. And then the S35 was the protein that was in the phage and it got knocked off, okay? And so the only thing that got transferred into the bacteria in this particular case, and it gets a little more complicated with other viruses, they were lucky that this particular virus worked exactly this way, but it gave them the correct answer, that in fact the radioactive proteins were actually left up in the supernatant, okay? And that wasn't what was being transferred over, okay? So, I'll set my mouse. Okay, well, let's go back now. Okay. All right, so, and the reason that works, right, was that it was in the DNA is actually what was going in, and that was ending up in the pellet. All right. The other experiment, and what's happening with these phages is that when they land on the bacteria, they actually have proteins that allow them to, the receptors that allow them to hook onto the bacteria. They inject their DNA in, it sits inside the cell, and if it goes lytic, meaning that it's going to kill the bacteria, then it reprograms that DNA carries the information to then make more viral particles. It also replicates its own DNA, which we'll talk a little bit about in further detail today, and then those phage can be released, okay? So it's the DNA is the source of the genetic information. The other example we gave you was this experiment from Griffiths, which was actually done much earlier, okay? But this didn't say what the material was. It just made that observation again that we were able to transfer some sort of information, in this case from dead bacteria to live bacteria, okay? And that information allowed them to actually be pathogenic and kill the mice, okay? So it turned out that there was this paper published in 1944 which indicated that the material that was actually being transferred was actually DNA. So it was a chemical, they could actually purify the liquid, and they could separate out the components, and they could show it was actually DNA that was the active material that was being taken up by the R cells, basically turning them into S cells, okay, so that they could kill the mice. Okay, so this is what we went over before. The only difference is that now I'm telling you that chemical analysis was done, and it was shown that was actually DNA. The interesting thing is that this experiment was actually done quite a bit before the phage experiment, but people largely seem to ignore this experiment for some reason, and the phage experiment is what became more famous, probably because phage geneticists were just more popular than the people working on bacteria at the time. But now we know a little bit about how this actually happens. So if we break open a bacterial cell, a bacterial cell normally just has a single it has a single circular chromosome of DNA in it, and so that comes spilling out. It's giant. It's all looped around here. But if you look, there's these smaller circles of DNA that come out that are called plasmids, which are, again, just small circles of DNA, and they also replicate each time the cell divides. Okay? And they're in there. And it turns out that quite often bacteria have these small plasmids, and that they are. Show you that. Hold on. Okay, so quite often bacteria have these plasmids in them. Okay, but they actually can be transferred sometimes from bacteria to bacteria. So bacteria actually do have sex. It's a very modified form. They form a little connection between two cells, and then things like this plasmid actually can be replicated and make a copy of themselves, enter into the other bacterium, and then you have two copies of that. And this usually occurs in bacteria that are closely related to one another, or even the same species. But occasionally it occurs between very distantly related bacteria, and so they can pass they can pass plasmids between one another. Okay, and so this actually is one of the things that causes real problems with antibiotic resistance. So often the genes that confer resistance are spread because they're on plasmids, and they spread from bacteria to bacteria by conjugation by this process of these connections between bacterial cells. Okay, and so what would happen, it's not that when you're infected, if you have some sort of infection, you take penicillin, and then you end up with a problem because the bacteria you have are resistant. It's not that the idea that there's a resistant bacteria and it's spreading it at that moment into all the other bacteria in your body. What's happened is that if you have a few resistant bacteria, you may be able to kill 99.9% of the bacteria that are infecting you with penicillin, but if there's 0.1% that for some reason have a plasmid that gives them resistance to penicillin, then they're okay, right? And they reproduce quite rapidly, and then you're still sick, okay? And so what happens, like in the case of penicillin actually comes from a fungus that lives in the soil. And so there are soil bacteria that have evolved long ago to be resistant to penicillin, okay? So the fungus uses it to stop the growth of bacteria. Penicillin doesn't actually kill bacteria. It actually stops them from building the cell wall properly, so they can't survive, or they can't continue to divide. And so some soil bacteria evolved resistance long ago. And the problem is now that we start using it as an antibiotic in humans, right? On rare occasions, then, the soil bacteria have been able to transfer the plasmid containing resistance to penicillin into bacteria that are pathogenic in humans. Okay, and now they then spread to each other over time. And so lots of bacteria that we used to be able to treat with penicillin, we no longer can because that soil plasmid has basically gotten into all these other bacteria now, and they can pass it by conjugation. Okay, and so that's what happens, and that's one of the things that plasmids do. Okay, and so it turned out that it looks like this is some sort of DNA-based system like this, that in fact the S cells and R cells are the same species. They have the same bacterial chromosomal DNA, but the S cells have a plasmid. Okay, and what's happening is that when you heat kill the cells, the plasmid leaks out. Okay, and then when you mix it with R cells, those plasmids can enter into the R cells. So some bacteria are actually very good at just taking up DNA from the environment as well. And so that happens, and that's why then these cells become pathogenic. So what do you think that the plasmid might encode? What's that? Okay, so it talks, but actually, what do you see the difference between the S and R cells? Yeah, it's this coat. So the coat is what allows them to be pathogenic, and that's what's encoded on that plasma, on that piece of DNA, is the gene for making that coat. And so when C cells get that plasmid, then they can make a coat, in fact. And then, of course, all this bacteria that come out of there, you would consider S-type bacteria. They now have that plasmid, they now make that coat, okay? And they're able to kill. In reality, just to be honest with you guys, is that it's a bit more complex. In this case, the DNA is not really a plasmid, it's a free piece of DNA. It turns out that this type of bacteria is very good at just taking up little fragments of DNA in the environment. And so that DNA was actually in the chromosome, and it just got spewed out. And it only has to happen very, very rarely, okay? If you have, 5 million bacteria, if one of them is able to take it up, okay, then this is going to be a problem, because that one then in a day can divide lots of times, and you'll have a serious infection. So that's the idea, okay? All right. So again, normally though, most of the DNA that's replicating in a cell is the chromosomal DNA. So in bacteria like E. coli, it's actually a giant circle, but it's wound up into sort of a knot-looking thing. And when bacteria divide, one of the things they have to do is they have to replicate all that DNA. And so what we're going to do for most of today is talk about how DNA replication occurs. Okay? And it's a pretty complicated process. And so I'm going to follow along with a textbook, and you can go back. I encourage you to go back to the textbook and go through it yourself. But I'll go through the whole process. So when bacteria divide, what happens is that they initiate the replication of the DNA in a particular spot, and we'll talk about this. They have these what are called origin of replication, and those two origins then go to opposite ends of the cell. And then when the cell actually divides, each cell gets one copy of the DNA that had been in a single cell. Okay? And that's a pretty straightforward concept, that when you divide a cell, the two daughters need the same information. In this case, it's DNA. So they replicate the DNA. They have one copy to two copies, and then they make sure to segregate that faithfully between the two cells. And in the next lecture, we'll talk more about how the other aspects of cell division work. But today we'll just talk about how they replicate their DNA. Okay? And so then they divide, and you get two daughter cells. Okay? So you remember what the structure of DNA looks like. So you learned about it in the first third of the course. And so this is the way we normally picture it, as this helix, right? And so Watson and Crick, using lots of various lines of evidence, including the X-ray diffraction data from Rosalind Franklin, actually figured out what the structure of the DNA was. And they realized that it's a backbone that's made up of sugar and phosphate, right? And then these nucleotides that are facing inwards into that helix. Okay? And it's a double helix running in what we call anti-parallel direction. So one strand, we can label these as 3' and 5' ends based on the direction that the phosphates connect to the sugar. Okay? And so they run in opposite directions to each other. And by convention, we call this the 3' end and the 5' end. So the 3' end is the hydroxyl, the 5' end is the phosphate. And so the strands the sugar backbones run in opposite directions here, and then the nucleotides are in between. Okay? And then the whole thing is a helix. There's about 10.5 base pairs per one turn of the helix. All right? So it's a helical structure like that. And the reason that solving the structure of the DNA was so important is that it immediately gave rise to a model as to how you would duplicate it faithfully. Okay? So remember again that the base pairs pair in a very specific way. Okay? So you always have A with T and G with C. Right? So you guys went over that before, right? So you had that. And the reason for that pairing is precise, is twofold. One is the hydrogen bond pattern. Okay? So this pair makes 3 hydrogen bonds, this pair makes 2. But if, say, an adenine tried to pair with another adenine, the problem is that those are two relatively bulky nucleotides, and the spacing would be wrong, and they would push apart the double helix. Likewise, if you had one of the smaller ones, like a cytosine, trying to pair with a thymine or with another cytosine, the problem would be is that the spacing would be too small, and it would also disrupt the helix. So it's only these pairs that work in DNA. Okay? And so the structure lent itself to an easy explanation for how you would do this duplication. So the key was they realized it was a double helix. Okay? And other people, like Linus Pauling, who was working on the structure at the time, thought it was some sort of triple helical structure and things. But the double helix makes sense because what happens is here are your two strands of DNA, okay, that together make a double-stranded DNA molecule. If we take them apart, okay, like that, okay, and we're going to have the old strand in dark blue, and what's going to happen is that nucleotides can come along, but they will match, right, in a precise way. Okay? And now if you look at this, you started with this sequence here, this molecule, and you split it in two, and then you let the nucleotides come in and line up. And what you notice is now these two are identical copies of this. Okay? And so that was the key. That let you take a strand of DNA, which has 4-letter code, but like in you, right, your DNA sequence is something like 3.2 gigabases. Okay, so it's 3,200 million nucleotides, or billion nucleotides. So it's this enormously long, well, it's broken up into a couple chromosomes, but it's a huge amount of information that has to be faithfully reproduced each time. And so this is part of the key, is that because of this chemistry, when the two strands are taken apart, the nucleotides are going to line up properly. Okay? And so what you have to do then is zip this all back up, make the bonds again, and get double-stranded DNA. And so we'll go over how that works. So one of the things that the people questioned was whether or not it really worked in exactly this way, meaning that you had the two parental strands here that separated, and then the new strands came in. And so the resulting molecules, the resulting two double-stranded molecules, had one strand that was from the parent, and then one strand each that was new. Okay? And then the idea was that each daughter would get one copy of this when the cell divides. All right? And so if you think about it, there are a couple things that could have happened when it divided. So and these are these three models called the conservative model, a semi-conservative model, and a dispersive model. So in the conservative model, what would happen is that it would obviously replicate, but that one double-stranded result was all new nucleotides, and the other one was all the old ones. Okay? Another model was called the semi-conservative model. The strands would come apart, and then there would be a new strand associated with each old strand, and then each daughter would get one of those. Okay? And then, would get a pattern like this, and then when they divided again, it would be this semi-conservative model where then old strand would stay together here, and old strand would stay together here, but then these two daughters would have these new strands. Okay? Does that follow? And then there's sort of this dispersive model where, in fact, bits and pieces could exchange, and so you would actually have a mosaic of old and new pieces of old and new nucleotides in these double-stranded molecules. Okay? So, yeah. Yeah. So, sorry, it would be the same as it. Oh, so it just means that there are new nucleotides and old nucleotides. Okay, so the sequence of these two molecules would be exactly the same, but these would all be newly synthesized nucleotides, and these would all be the old nucleotides from that strand. Okay, does that follow? Yep. Sorry, which one here? B? Because, okay, so now if we have the same model, okay, so in essence what would be nice is if they made 3 shades of blue, but the idea is that if you label all of these original nucleotides, right, then everything here in each next round is new, and these two would be this light blue strand plus an even newer strand that was put on there. Okay, so they'd be, if you wanted to make it accurate, you would make this shade of blue and the other strand an even lighter shade of blue, because it'd be even newer nucleotides. Okay, so there was a famous experiment by Messelson and Stahl that solved this problem, and I'll go through it very quickly. It's not too hard to understand, but it's a classic experiment, which is that what they did is they cultured the bacteria in a heavy isotope of nitrogen, and again, that ended up in the DNA. Okay, and what they could do then is then transfer it into media that had a lighter isotope of nitrogen. So the idea is that in this phase, you make the initial strands of DNA. Okay, and now you're going to let the bacteria divide, but they no longer have the heavy isotope of nitrogen; they just have the light isotope of nitrogen. So again, this would be like the dark blue color and the light blue color. Okay, and then they could let them undergo 1 round of replication and 2 rounds of replication, and they would harvest them, and then they would analyze the DNA that they collected. And the way they analyzed it was to centrifuge it into a media that was into a substance that was very dense. And what happens is the DNA goes until it reaches the same density. So it's a gradient of density. And so what's going to happen is the DNA is going to centrifuge down until it matches the density of the material that it's in. Okay, and so the density is very low up here and high here. So what happened? What they saw is that initially they just got 1 band. Okay, so all the DNA had the same density after that first, and then it ended up having 2 different densities. One of those that was the same as this, and then another one that was lighter. Okay, so if you think about this, then that's consistent with the semi-conservative model. Okay, so let me tell you how that happens. So the reason you get a single band here is because now both of these are the same in that they're 50% heavy nitrogen and 50% light nitrogen. Okay, but they're both the same in terms of density, so you see that band. If you had this model, you would have gotten 2 different bands. One would be all heavy and one would be all light. Okay, but you didn't get that, so this is clearly not the right model. But these 2 models would give the same thing until you got to the next division. So in the next division, you ended up with one that was the same here, the same band, and then a light one. And that's consistent with this idea that you have the semi-conservative mode, and you would still have these 50-50 ones, and then all the other ones would be brand new nucleotides, all light. Okay, and that wouldn't happen if you did this. You would still get this kind of, you would cut in half the total amount, the ratio of heavy to light in the next division. You would still just get a single band, and it would be lighter than this one. Okay, so that was the classic experiment that showed that you had the semi-conservative model of DNA replication. All right, so what we'll go over now is a little bit more in detail of the sort of biochemistry of what happens when you replicate a DNA molecule. So the thing is that if something like a bacterium, right, you have this large circle of DNA, and in reality, of course, it's like I said, it's knotted up, but you can think of it as a circle. And what you're going to do is you're going to open up the circle at one point, and that's called the origin of replication. And then you're going to start lining up nucleotides and synthesizing them in both directions. Okay? And so if you think about it, it's this double helix. You pull it apart, and then you start copying it in both directions going around. And what you're going to end up with is 2 circles. Okay?